Hello everyone, welcome to 𝗲𝗵 in which we were discussing the general microbiology, and this is 11th class, the last class, and we'll discuss the general virology in today's class. So, giving an introduction to viruses, remember that viruses are acellular organisms. They don't have cells. Viruses don't have cells. But viruses have a unique property to grow up inside the cell. They are obligatory, compulsorily intracellular. They can't grow without your cell. You are there, then only they'll grow. Okay? So, viruses entering the human body and saying them, although my dear viewers, I am a singer also, right? So, I'll tell you what it says. The virus, when it comes into your body, meets your cell, what does it say? Tu hai to dil dhadakta hai. Okay? So, this is what is said by viruses to the cells because they are obligatory intracellular organisms. Okay? So, these viruses are seen only on the electron microscope. Why? Because they are nanometers in size. And no culture media, understand, no culture media can grow them because they are obligatory intracellular. They might grow on the cell lines. Okay? So, there are some cell lines specifically on which the virus materials can grow. You detect the viruses by using ELISA or PCR because it is not going to get stained. It is not going to grow in the culture media. So, culture media and stain, these all everything is for bacteria. It is not for the viruses. And viruses, because they can't grow, they can't be cultured, there is no way to attack them, right? So, the treatment options are also very less. So, only few viruses can be treated, and that's too, treatment is by the vaccination. My dear friends, remember the viruses are divided into some classes like DNA, RNA viruses, and that's too can be single stranded or double stranded. So, you have got single stranded DNA, single stranded RNA, double stranded DNA, double stranded RNA. They can be enveloped, they can be non-enveloped. Okay, so they can have an envelope outside, they might not have. Symmetry, either they will be helical, so this is a helical symmetry, or they will be icosahedral. I'll teach you in detail what does icosahedral means. When some particular example in systemic virology comes, I'll teach you there. Coming on to DNA viruses. So, the examples of DNA viruses are herpes, hepatitis, pox, 𝗵𝗵𝗵𝗺. So, I used to recite it like a poetry: herpes, hepatitis, pox, 𝗵𝗵𝗵𝗺. Okay. So, this is the way you have to remember: herpes, hepatitis, pox, 𝗵𝗵𝗵𝗺. And if you see carefully, APP becomes application. And you tell me, if you are giving a letter, a love letter to somebody, you will keep an envelope and give. But if you are submitting an application to the principal, will you keep an envelope? No, my dear friends. So, that's why application, app does not have any envelope. That means APP don't have any envelope. They don't have any envelope. So, non-enveloped out of them are 𝗵𝗵𝗵𝗺, whereas herpes, hepatitis and pox are enveloped ones. Who is single? I am single. So, single stranded DNA means parvo. Rest all are double stranded, herpes, adeno. Oh, where is hepatitis? So, hepatitis is partially double stranded. So, can you see the B and the P look same? So, that's why the partial one is the B one, the partial one is the hepatitis B. Let's quickly revise. Completely double stranded, herpes, adeno. Hepatitis, partial, single, I am single. Enveloped, herpes, hepatitis, pox. Non-enveloped, APP, parvo and 𝗵𝗵𝗵𝗺, 𝗵𝗵𝗵𝗺. Symmetry. So, remember brick is just like box. So, brick shaped, box shaped is pox. So, box, pox, brick, box, pox. All other are icosahedral. Coming on to RNA viruses. Again, single stranded RNA, double stranded RNA. Who is single stranded RNA? Rotavirus. Okay. Rotavirus is a double stranded RNA. Why it is double stranded? Double stranded. Okay. So, Who is the famous 𝗵𝗵𝗆 of India? 𝗵𝗵𝗆 is in actual sense is a little 𝗵𝗵𝗆. Okay. So, Rota, just like 𝗵𝗵𝗆, 𝗵𝗵𝗆 is having थोड़ी सी health, double हो गए, double, he is double. Okay. So, that's why rotavirus double stranded. Okay. You got it? Great. Single stranded, but jokes apart, 𝗵𝗵𝗆 is Jaan of our country. Thank you 𝗵𝗵𝗆 for everything. Okay. So, coming on to single stranded RNA. Okay, single stranded. Now, single stranded can be positive sense, can be negative sense. So, if you have a PCR machine, you can find the viruses. I am talking a little off the record that if you are having a PCR machine, you can find the viruses. So, once upon a time, all the viruses gathered together and take care if PCR machine finds you. Take care if PCR machine finds you. But mind you, all these viruses were single, or you can say that all these viruses were SRs. SRs means single stranded RNA. Take care, TC means toga virus, C for coronavirus, PCR, P for picorna, 𝗏𝗃, R for retro, just like HIV, finds you, 𝗵𝗲𝗃, just like hepatitis C. Okay. But what about negative sense? So, negative sense you have to remember, if you will think negatively, the BMW is far away from you. But if you think positively, you can own a BMW. So, BMW far away if you are thinking negatively. So, negative sense you have got far, FAR, F for philo, A for arena, R for rhabdo. BMW, B for bunya, M for myxo, that means paramyxo and orthomyxo. W is not there. Okay, W is just like to complete the mnemonic. And paramyxo family has got measles, mumps and respiratory syncytial virus, MMR. Generally in MMR it is rubella, but here it is not rubella, it is RSV, because these three are the example of paramyxoviridae. This is very important, please don't forget. MMR is an example of, measles, mumps and respiratory syncytial virus is an example of paramyx. Okay. Coming on to segments of the RNA viruses. So, the RNA virus is also having some segments. How to remember? Some segments means, okay, I'll make this diagram. So, like this, 1 segment, 2 segment, 3 segment, okay, some might have 4 segment, so like that, okay. So, how to remember how many segments? So, you must be aware of alphabets and then you can easily remember the number of segments lifelong, just like A. Are you aware that A, B, C, D mein A, in A, B, C, D, A is the first letter. Just add 1, A plus 1, A is the first letter. So, add 1, that becomes 2. So, A, that is arenaviridae, has got 2 segments. Similarly, you have got B. So, B for bunyaviridae. So, bunyaviridae has got how many segments? B plus 1, 3 segments. Then next is orthomyxoviridae. So, O down, O up become 8. So, 8 segments. Reoviridae. Reoviridae, you have to remember 10 segments are there in reoviridae. Okay. You got it? Okay, let me write my version of 10. So, this is how I write 10. If you just put like this becomes reoviridae. Sometimes you must be laughing on me now, oh my god, how foolish mnemonics, but believe me, this is the only way to remember microbiology. Okay. Now, non-enveloped viruses. Non-enveloped, once I have told you, APP is the mnemonic, right? Adenopapovaparvo. Okay, so APP I have told you, but the full mnemonic is actually 𝗃𝗃. So, 𝗃𝗃 is CRAP, that is calci, rio, astro, picorna, and then APP. Okay. So, these are the non-enveloped viruses. Rest all the viruses in the entire world are enveloped viruses, enveloped viruses, enveloped viruses. Okay, you got it? Helical. So, helical, please remember, one of the lady chief ministers of India has been Rabdi Devi. So, RBCM. So, CMRB. Okay. So, Rabdi Devi was standing for CM candidate. So, RB. So RB means R for rhabdo, B for bunya, CM, C for corona, M for myxo, right, and F for philo. Okay. So just remember that Rabdi Devi for CM candidate for 6 years. Why I am telling you 6 years? Because helix is just like hexa. Okay, so 6 is hexa, and hexa you can remember helix. Okay, rest all are icosahedral. Okay, rest all are icosahedral. Coming on to how do the viruses multiply. So if you want to learn about the multiplications of viruses, let's just understand one by one. First of all, please understand that you have got the very first thing is the attachment to the host. So this is the host, this is the virus. The virus is going to attach to the host. How? With the help of some receptors. Just like coronavirus gets attached to ACE2 receptors in the respiratory epithelium on the respiratory mucosa. So attachment to the host cell. Once it is attached, it is going to enter. But entry occurs only with the help of nucleocapsid, not the envelope. What does that mean? See here, if I just make a whole virus, this red one is the virus and this green one is the envelope. So what happens, in order to enter inside the host cell, it is going to remove this green color envelope, and the virus without the envelope is called as nucleocapsid, which I have made from the red color here. So this nucleocapsid with no envelope is going to go inside the host cell. Once it goes inside, now it is going to release the nucleic acid, and once it releases the nucleic acid, this is called as uncoating. And once the genetic material is outside, the genetic material now is going to multiply. Multiply DNA viruses, multiply RNA viruses. So DNA viruses are also going to multiply, RNA viruses are also going to multiply. Now please understand the beautiful story. DNA is dominating. See here, DNA, D for dominating. So DNA says, I am dominant than you RNA, shut up. So DNA says, I am dominant than you RNA, shut up. I am not going to leave the nucleus, I will multiply inside the nucleus. RNA says, okay. So DNA is dominating, that means all the DNA containing viruses are going to multiply in the nucleus. Whereas RNA has to go to cytoplasm. So all the RNA viruses are supposed to go to cytoplasm. But exception is there. Pox has a love for cytoplasm. Who is the lovely, cute, beautiful actress who has left India, went abroad, married to 𝗲𝗵 Jonas? Yes, Priyanka Chopra. So remember, P for pox, C for cytoplasm. So pox goes to cytoplasm. Okay. So pox is a DNA virus, but it goes to cytoplasm. Whereas please remember something which is close to the heart of all the Indian mothers, that is the Rin bar. That's a soap washer, Rin bar. So Rin means all the RNA ones are going to cytoplasm, but influenza, which is an RNA one, has to go to nucleus for its replication. So Rin bar and Priyanka Chopra, both of them, please don't forget. My mother's favorite, Rin bar, my favorite. Okay. Coming on to the release. Now, since they have replicated, so many copies have been formed. Come back to the topic. Don't go to Nick Jonas and Priyanka Chopra. So once it has to release, how it will release? Enveloped viruses. The viruses whose genuine nature is enveloped, they are going to release by budding, whereas non-enveloped viruses are going to release by lysis. Why? Because I told you, see here, this was the one, yeah, so they have multiplied now and made their copies, okay, made their copies like this, outside although it was enveloped. So whenever they'll come outside, because the genuine nature of these viruses are that they are enveloped only, so they will carry the blue membrane of the host along with them, like this. in order to call themselves as enveloped ones. So how will they release? So they will release by budding. So one bud, then second bud, then third bud, so that envelope can also be formed with the help of cell membrane of the cell. So that's why enveloped viruses are going to come out by budding, whereas non-enveloped they don't need. So they will just destroy the cell and come out. That's why the envelope has got two layers. The inside is for made by virus, obviously that's a true layer, and the outside by the host. Okay, so the envelope is made up of lipid bilayer, the progeny's envelope. Okay, coming on to the identification of viruses. In order to identify different viruses, because I have told you that there are only, very less ways available to identify the viruses. So how will you identify? See, these viruses are very special. They are going to exert some special effects on the cells, and we label these effects as cytopathic effect. Cyto means cell, pathic means pathology. Pathology means abnormal. Okay? Abnormal effects. For example, CMV, 𝗖𝗵𝗵𝗺. Cyto means cell, megalo means increase. So 𝗖𝗵𝗵𝗺, CMV. They do, cyto means cell and megalo means ballooning. So CMV causes ballooning of the cell. So in a histopathology, if you are watching ballooning of cell, you can suspect CMV. Very good. Adeno. Adeno word, wherever comes, you have the gland associated with it. Adeno means gland, and glands secrete, and secretions, they are going to form the cluster. So wherever you can see clusters inside the cell, this is the, clusters of the cells, you can understand it is adenovirus. Polio causes lysis of the cell, POLLY. So just like you can, LI is for what? Lysis. Okay. Then syncytia. Syncytia means this was one cell, another cell, both the nucleus come together and form a single cell. This is called as syncytia. So the respiratory syncytial virus is like that, and even HIV is also like that. Sir, how to remember, sir? Oh, very simple. How does HIV spread? One individual, second individual, HIV. So this is the way to remember how the HIV is spreading. And then comes the measles, herpes, focal degeneration. So MDH is a very, if you know, that's a very popular brand for spices in India, and if you don't know, please Google it. This is very popular. So MDH is a spices brand, D for degeneration, M for measles, and H for herpes. Some people might say, oh sir, we don't know this brand of spices, how will we remember? But it's okay. It is easy to remember MDH rather than measles, herpes and focal degeneration, or you can make your own mnemonics also, I don't have any issue. So this is one way to find out that which virus has attacked the body. Now we will understand about the second way, inclusion bodies. There are some bodies which are specifically seen with the specific viruses. For example, Negri bodies. These are seen in rabies. Can you see? Negri has got a word R and I, and rabies, right? So Negri, the RI starting the rabies, Negri bodies. Coming on to 𝗏𝗏𝗃 B, I have intentionally skipped 𝗏𝗏𝗃 A, come on to 𝗏𝗏𝗃 B. I have told you that B, A, B, C. So A for adeno, B for 𝗏𝗏𝗃 B, and C for CMV. And B and P look same. So polio also has 𝗏𝗏𝗃 B and inclusion bodies. 𝗏𝗏𝗃 A has got HSV1, HSV2, varicella zoster 1, yellow fever, molluscum contagiosum. So do you remember molluscas having octopus, yellow color? Yes, yellow color, okay, and having 8 limbs, so heavy ones. So remember, heavy, that means H and V, HSV1, HSV2, V for varicella zoster viruses, and then you have got mollusca, and then you have got yellow fever, mollusca and yellow fever. Okay, coming on to guarnary bodies for variola and vaccinia. Guarnary bodies for variola and vaccinia. This you have to remember. Okay, and Bollinger fall. So B and P go together, B and P go together. So you can remember like that. Okay. Coming on to the inclusion bodies. So all these inclusion bodies were intranuclear. These were found inside the nucleus. But there are two organisms which are going to form it intranuclear and intracytoplasmic both. And because they are going to form intranuclear plus intracytoplasmic, we call it as combined ones. So my mnemonic is combined, COM. So C for CMV and M for measles. That means it is formed inside the nucleus as well as inside the cytoplasm, both of them. Okay. So yes, this is all about the general virology. We are done with the general microbiology also. Since the viruses are unique, they are not going to take the stain. They are not going to take the negativity of other people. Okay. Similarly, be like viruses. Okay. Stay in close contact with the good people, with your parents. Okay. Don't take what other people say to you. Stay away from negative staining. And this is the only take home message. Even if you forget general virology, it's okay. But don't forget that you should not give the remote control of your life and emotions to somebody else. Don't let other people hurt you. Even if they are hurting you, you should spread positivity. Because if somebody is hurting, he or she himself inside is very much in hurt. With this, Baba's Gyan is done for the general microbiology. I wish you all a very, very, very best of luck and God bless you. Thank you so much.